Thank you so much, Linda Gale and your co-chair for this invitation to participate. So these are short talks, so I wish we could talk a whole day about this, but I will try and keep it short. So the title was quite long, Innovative Clinical Trial Designs to Advance and Accelerate Increase in PrEP Choices. And I think when we talk about combination prevention, as you just did, then also choices in terms of availability of products that really meet the needs of all. And what might be good for one population in the US may not be the best product for people, for example, young women like Brenda was talking about in Africa. And I want to say that what I'm talking about today, these are my own views. I'm not speaking on behalf of the whole Forum for Collaborative Research, certainly not on behalf of any regulators, but obviously they come, what I'm talking about today is informed and influenced by some of the conversations we've had in the forum. And I came across, as I was thinking about this talk for today, I came across a slide that I used in 2007. This was a meeting in Kampala, Uganda, where the World Bank had asked me to talk about the role of research and program sustainability. Now think back to 2007 if you can. There was a lot of talk within PEPFAR, and people argued about should it be operations research or implementation research. Remember that? And it all came from engineering anyway, so what did it have to do with HIV? But in those days, programs and research were clearly very separated. The NIH only did research, no clinical, no therapy, no taking care of patients, and that was very clear. That's the NIH's mission. And programs were there to treat people and not to do research. So what we talked about at the symposium is that there needs to be some integration of one with the other because we need research for program sustainability. But now we're talking about how can programs inform research as well. And I came up for that talk with two different models, and I kept the slide as it was because I still have operational research in there. But obviously there's a lot of program evaluation going on in the programs. And sometimes, you know, there may be a little bit of a blurry line, as if I tried to show there, between what is a program effort and what is a clinical research effort. And then in the bottom is a more distinct way of doing it, where you can embed clinical trials within a program setting, such as, for example, doing community-based randomization. But in any case, we really need to think about moving forward is how we use all of the tools we have available to move forward. And when we talk about trial designs for PrEP, you know, forget about placebo, we can't do that. The standard of care, of course, we have several drugs now that have been approved, and not all of them in all areas of the world. And we can do a superiority or we can do a non-inferiority versus standard of care. For superiority, we would still need a standard of care. And what I'm trying to show on the right side here is that as we move through a timeline of research, we start here with the first, so this would be Truvada, for example. So that's intervention A, and we need a certain number of people to show the difference. But you can see that first time you test it, there's a huge difference, or there could be a huge difference, a treatment effect, between being on active versus being placebo. But once you have that, you maintain that effectiveness, so now your new intervention has to be better than that for a superiority trial. And as you continue then, so A versus B, then B versus C, this becomes smaller and smaller, and becomes harder and harder to do the research because it takes more patients, more participants to show a small difference and show significance. And of course, we also have fewer transmissions. So for all those reasons, that those trials, as I tried to show down here, get bigger and bigger. The N is getting much, much bigger than the original. And now we have intervention X, whatever we're going to test next, and how we're going to do this then. And so what we've been talking at the forum a lot is about how can we use a an external control instead of a placebo. But what I want to point out here that, and not, you know, sometimes it's hard to grasp this, whether you use an external control outside of the trial or a traditional placebo, both of those are actually counterfactual estimates of what would have happened in the absence of intervention X. But of course, if we use an external control that's not part of the trial, we lose randomization, and we always ask ourselves, is that external control population similar enough to the population that we are testing? Now, if we keep going along this research timeline, there comes a point where this difference between the new intervention X versus the control, the standard of care, it becomes so small that you may as well do a non-inferiority study, or you kind of have to because you can no longer show superiority. But what's really important to remember here is that a non-inferiority is basically saying your new intervention X is more or less as good as your standard of care, but that really is dependent also, it's referring back to your original placebo, because the efficacy of intervention C is based on the stepwise demonstrations of efficacy, but they all relate back to this placebo. Intervention A was better than placebo, and so intervention B is better than intervention A, but that's really significant because intervention A is so much better than the placebo. So we're always going back to that placebo. So it does in fact become, you know, it's a historic control in a way because it was many research generations ago. And Deborah Donnell actually gave a very nice presentation about this yesterday at our symposium. So when we then talk about a counterfactual where we actually use concurrent data from the communities and build a counterfactual estimate, you know, we'll have different values. They will not all be the same. But this is more concurrent to the intervention X and the standard of care compared to the placebo, which really requires for a non-inferiority trial that you meet the constancy assumption, which is very difficult to actually justify. So when we talk then about building a counterfactual estimate for HIV incidence using external controls, we can, for example, use an HIV recency assay, so that those who test HIV positive, we can then estimate how many of them, not individually, but at a cross-sectional population level, we can get an estimate of recency. And I really want to emphasize that this should also be supplemented by other data sources like surveillance or previous studies. So then we have this counterfactual estimate, and the primary comparison then is that the efficacy of your new candidate prep product to this counterfactual. And of course, you also have that other comparison that will strengthen the study. So this is the approach that is actually moving forward. We know that there are 2 programs out there that are applying this model. And, but basically, it's to use a counterfactual estimate for HIV incidence. More than one method should be used. And we already talked about the primary analysis. Now, what's important here? It's important that as these new PrEP products are being studied, one of the programs is still stopped, but we hope that it will resume in the near future. So they're testing this approach. And we as a community, through collaboration, need to monitor, evaluate, and course correct as needed, of course, because it's a new approach and people have a lot of questions, and they should. And we as scientists and communities really need to do our best to really follow closely how this is working out and what we can learn and what we can improve. But I really want to also applaud the regulators for their flexibility in allowing these programs to go forward in that way. Now, at the beginning, we talked to prepare for the long game in terms of next generations of PrEP, but notice that I've emphasized the S here, and it's really prevention research. Let's not just think about the 2 current programs. Let's not just think about maybe the next drug. Let's think about 10, 15 years into the future, and let's prepare for that as a community so that we know where we're going and we know what we need to do. And if we then go back to this model of what's happening in programs, what's happening in surveillance and public health, and what's happening in what's traditional clinical research, really find ways that we can have information from programs, help us estimate what the true incidence would have been. So what's important here? We need sound science and evidentiary standards. And again, I want to emphasize that we are not just talking about ARV-based PrEP. A lot of talks about broadly neutralizing antibodies here, and of course vaccines. And so with ARVs, we have decades and decades of experience of how they behave, how they reduce virus replication, etc. So we know how they act and what they do. And we've seen that over and over again. And every antiviral we have tested that has sufficient drug levels at the right sites has proven effective. So we come in with a whole set of already knowledge. When we talk about broadly neutralizing antibodies and vaccines, there we have less evidence. So we really need to think about what are the evidentiary standards in these different types of approaches. We need the appropriate ethical frameworks because we know ethics for clinical research, but now we're mixing clinical research with public health and surveillance, and we need to figure out exactly what does that mean and how will that affect the communities. And of course, it always needs to be community-based, and we need to ensure that we have benefit for the local and global communities. And with that, I thank you.
Thank you, Veronica. Great job.